This conference will now be recorded.
Hello students, I'm Dr. Divya from TeenEinstein. So already we have done with chapter 1. We have discussed the main points and even the previous year questions also. So now we are moving to the second chapter, biological classification. So we know that our exam, NEET exam of 2024, it's very near, and only 2 to 3 months we have to prepare for that exam. So we need to prepare well. So first, what I'm telling, read the chapter, carefully read the chapter. Each and every point should be there in your mind. And if you're not satisfied with your preparation, repeat that, and you have to find out the time. And main thing, after preparation, after preparing of each chapter, you need to note down which portion is difficult for you and where you are finding that difficult. You need to note down that. Okay, and you have better to make your own notes, like at least short notes, to make your own notes and be prepared for the exam. Each and every chapter is important, but when you're going for the exam, at least the portion which is having more weightage, that you should learn well. Okay, so if you're preparing, if you're planning well, it will be easy for you to crack this exam. So don't worry, you can clear this exam very easily. So what you need to do is, if you're sitting for one chapter, to prepare one chapter, completely concentrate into that point. In between, you should not go for anything. Your concentration should be 100% into that. Okay, so okay, let's move to the chapter 2, biological classification. So here also, as we did in the previous chapter, the same thing we are going to do. We are covering the main portions and main points we are going to discuss here, and we are moving to the previous year questions. Okay, so first, I'm going to tell you what is the syllabus, which portion you need to cover from this chapter. See, biological classification, actually, this is a very important chapter, right? Because here you will get so many confusions in this chapter. While reading this chapter, you will get confused. Okay, the first chapter was very easy, but this chapter is not like that. Here you will get many confusions or confusing points will be there. Biological classification. Okay, so first we are learning about 5 kingdom classification in this chapter, and the features and the classification of each kingdom, and what are the main features. So here, features and classification of Monera, Protista, fungi, and all these we are learning. Then we are learning about lichens, viruses, viroids. So here the 5 kingdom classifications we are learning, and we are mainly learning about the detailed study will be about the Monera, Protista, and fungi. Other 2 kingdoms that will come later. That will be a complete chapter for you. Plant kingdom you have to learn separately, and animal kingdom also you have to learn separately. So that will be a vast chapter for you. So here, what we are going to do is we are learning why classification is done and how it is done, and kingdom Monera, what and all the features you need to learn from that, and kingdom Protista, kingdom fungi, all those things we are going to learn in this chapter. Okay, so let's move to the chapter biological classification. See here, what is biological classification? That's the first question. Okay, so we need to tell very clearly that biological classification means that is the scientific procedure. In a scientific way, we are arranging the organisms into groups and to subgroups. Okay, it's a scientific procedure. Scientific way of arranging organisms into groups and subgroups. Okay, so that's how we are doing the biological classification. We are classifying the organism by using scientific methods, and then we are making them into groups and subgroups. That is the biological classification. Okay, so on what basis we are doing this classification, or how we are classifying? So we are classifying, or we are making the groups and subgroups on the basis of their similarities and differences. Okay, this is based on similarities and differences. Okay, that's the way of classification. Based on similar organisms will group into one, and the dissimilar one will make to another one. So based on similarities and dissimilarities, we are classifying, and we are placing that groups in different categories. That hierarchy of classification, that is based on this grouping system, or the method of this grouping. And this grouping, how we are doing, that is based on the similarities and differences. After grouping, placing them in categories. Okay, so that's how biological classification is done. So there are three main systems of classifications. Okay, three main classification systems are there. Which are the three principal systems of classification? One is artificial system of classification. Artificial system of classification. Second one is natural system of classification. Natural system of classification. Then the third one is phylogenetic system of classification. Phylogenetic system of classifications. So there are three principal systems of classifications. Which are they? First one is artificial system of classification, second one is natural system of classification, and the third one is phylogenetic system of classification. So these are the three main systems of classifications. Okay, so what is biological classification we need to learn, and based on what? Based on similarities and differences that classification is making, and we are placing them into different categories. So mainly three classification systems are there. One is artificial system of classification, second one is natural system of classification, and third one is phylogenetic system of classification. Okay, so let's see what is this artificial system of classification. What is this artificial system of classification? See here, artificial system of classification means that is like based on a few external characters. Okay, based on external characters. So which and all external characters, like few external characters they are choosing for the studies, and which are the external characters? First, like example, we can say morphology, morphological features, then their habit, then habitat. So all these features they are selecting for the classification. So this artificial system of classification includes study of few external characters. So that is based on the external characters. Few external characters they will select and they will study that, and based on that they are making the classification. So external characters in the sense morphology, then habitat of that organism, habit of that organism, everything we are considering, and then they are making the classification of that organism. Okay, so in this classification, Aristotle, then Linnaeus, these people are the pioneers of this artificial system of classification. See, Aristotle and Linnaeus, they started this artificial system of classification. So they are the these Aristotle, Linnaeus, these people are the pioneers of the artificial system of classification. And the artificial system of classification is based on few external characters like the morphology and habit of that organism. Okay. So this is artificial system of classification. Next, let's see what is natural system of classification. Second one we said natural system of classification. See here, first you need to know why we need to classify, because it's not possible to study each and every organism. It's impossible, right? That many organisms are there in this world. So we need to classify the organism. See, we said in the previous chapter, we said that is helping us to find out any new organisms are evolved or not, right? Not the one organism which is not including in any of this classification means it's a newly developed one or newly evolved organism, right? So evolution is a continuous process, so new things will get evolved, so we can find out the new organisms, right? And we can include them into a new category, so that we can do. That's why we are classifying the organism. That is the necessity of the classification. Here biological classification, that is based on the similarities and dissimilarities. We are making them into groups, then we are placing them into a right category, right? So that biological classification is of different types. Artificial system of classification is there, which is based on the external characters. Okay, and Aristotle and Linnaeus, those people, like, they are the pioneers. We need to mention they started this artificial system of classification, right? Second one is natural system of classification. So this natural system of classification, that is based on the studies of similarities, both the external and internal observable features we need to consider for this. Okay, so natural system of classification is based on the, it's based on the similarities. So we need to conduct studies on the similarities. Similarities in the sense both external and internal. External and internal features we need to consider. But both should be observable. Observable features based on the similarities, and that similarities should be observable. We can consider the external similarities and internal similarities, but the observable features only we should consider. Okay, that system of classification, which one? This natural system of classification was given by Bentham and Hooker. Bentham and Hooker. So Bentham and Hooker in 1863. In 1863, Bentham and Hooker started this, or they introduced this natural system of classification. So natural system of classification, it is based on the similarities. That similarities may be external similarities or internal similarities, both we can consider. But that feature which we are considering, that must be an observable feature. Okay, so who introduced that? Bentham and Hooker introduced this natural system of classification. Next, the third one. Third one is phylogenetic system of classification. Phylogenetic system of classification. Here in this phylogenetic system of classification, phylogeny means what we have already learned. It's a study of evolution. So this classification, phylogenetic system, we are following which system? Phylogenetic system. So we are following the evolutionary history and the genetic relationship of that organism. It's based on the evolutionary history and genetic relationship. Based on the evolutionary history and based on the genetic relationship of organism. That means the phylogenetic relationship. Based on the phylogenetic relationship, we are doing this classification. Okay, and this phylogenetic system of classification was given by Engler and Prandtl. Introduced by Engler and Prandtl. Okay, and this is in 1887. So phylogenetic system of classification is based on the phylogenetic relationship, or we can say the evolutionary history of an organism and the genetic relationship. Based on that, classification is doing means that is the phylogenetic system of classification, and this was introduced by Engler and Prandtl in 1887. So there are three systems, main systems of classification. First one is artificial system of classification, second one is natural system of classification, and third one is phylogenetic system of classification. Artificial system of classification is based on the external characters, like morphology, then the habitat, all those things. Okay, and Aristotle and Linnaeus, they introduced that system. The natural system of classification is based on the studies of similarities. That similarities may be external or internal, but that must be an observable character or feature. Okay, and this system of classification was introduced by Bentham and Hooker. And the phylogenetic system of classification, which is the third one, that is based on the evolutionary history and the genetic relationship of an organism, and this was introduced by Engler and Prandtl. Okay, so these are the three main systems of classification. Okay, next we are going to learn about the five kingdom classification. Classification is done, right? So that based on that classification, five kingdoms, they proposed five kingdoms. They made five kingdoms and they classify the organisms into these five kingdoms. Okay, so the most acceptable classification system, this was proposed by R.H. Whittaker. Okay, so five kingdom classification was proposed by R.H. Whittaker. Whittaker. Okay, and this classification was proposed in 1969. So this was in 1969, and who proposed this five kingdom classification? R.H. Whittaker. This was the most acceptable, or the accepted classification, we can say. Most accepted classification, and this was proposed by R.H. Whittaker in 1969, and this classification is also called, as we are calling that classification, the most accepted classification, as five kingdom classification system. Okay, this is called as five kingdom classification system. Why? Because Whittaker classified the organisms into five kingdoms on the basis of several reasons. Okay, so first we will say the classification. Organisms, they're classified into five main kingdoms. Okay, and the organisms are classified into five kingdoms on the basis of complexity of cell structure. Okay, based on the complexity, the reasons for this classification, why the organisms are classified into five different kingdoms. So these are the main criteria which he considered for the classification. Complexity of cell structure. The cell structure is complicated means they will go to the higher level of classification. So the complexity of cell structure is the one feature which he considered. Then thallus organization. Thallus organization. Then third one is mode of nutrition. Mode of nutrition. Next point is ecological lifestyle. That we can include the mode of reproduction also. Reproduction. Okay. And the fifth one is phylogenetic relationship. This is one feature for the classification. Okay. Then these are the main characters or features which he considered for the classification. So which are the main features? Complexity of cell structure, thallus organization, then their mode of nutrition. Based on mode of nutrition also they are classified. So ecological lifestyle also considered like mode of reproduction, then the phylogenetic relationship or the evolutionary history also he considered as a feature for this classification. And this is the most accepted classification, and this classification was proposed by R.H. Whittaker. This is also called as, see, by considering all these features, see whatever I have mentioned here, he considered all these characters for the classification, and based on these features, R.H. Whittaker classified the organisms into five major kingdoms. That's why it is called as five kingdom classifications, and this was the most accepted classification. Okay, so he considered phylogenetic relationship, thallus organization, mode of nutrition, complexity of cell structure, then ecological lifestyle, like mode of reproduction. All these features he considered, and he made the organisms into five major groups, and this was proposed by whom? R.H. Whittaker, and organisms were classified into five major groups. Now let's see which are these five major groups. So the 5 kingdoms of classifications, which are those kingdoms? 5 kingdoms. Let's see. First one is kingdom Monera. See, this is the first one, basic one, or the starting level one, kingdom Monera. Next, little more higher, right, based on the features. This is showing the basic features, little more higher features. Here, kingdom Protista. Protista, the second one. Okay. And the third one, that is kingdom Fungi. Third one, kingdom Fungi. Little more advanced features that was showing that king members of those kingdom it was showing. Then fourth one is Plantae, kingdom Plantae, the fourth one. Okay. And the fifth one is kingdom Animalia. Fifth one, all animals are included in this, and kingdom Plantae, all plants are included in this. Fungi, then Protista and Monera, based on the basic one. Those are coming in Monera. Then comes the advanced ones are coming in the higher ones. So Protista, the next one is Fungi, then Plantae, then Animalia. So these are the 5 kingdoms of this classification. Which are the kingdoms? Kingdom Monera, Kingdom Protista, Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia. Okay, so these are the major kingdoms, or the main kingdoms of this 5 kingdom classification. Then now we need to learn about each kingdom in this. Okay, about each kingdom we need to learn, because you may get several questions from this, and you need to note down all these points, because you will get confused about the features, right? Kingdom Monera means organisms like bacteria. Those are coming under kingdom Monera. So whichever points, all the points whatever we are writing, we should have a clear picture, a clear idea about that. Okay, we are, because next we are going to kingdom Protista, so don't get confused. They'll give, you'll get questions like they'll give an organism, this is coming under which kingdom. See, whether it is a plant or animal, you can differentiate it very easily, right? So don't get confused about this organism. That's why you should know the features. Okay, so here kingdom Monera, generally we can say bacterias. They are the sole member bacteria, we can say, and this kingdom contain 2 major groups. Okay, so this kingdom Monera is having 2 major groups. Kingdom Monera is having 2 major groups. Which are the 2 major groups? First one is Archaebacteria, and second one is Eubacteria. Okay, so these are the 2 major groups of kingdom Monera, Archaebacteria and Eubacteria. Okay, so don't forget kingdom Monera consists of 2 major groups. Which are the 2 major groups? Archaebacteria and Eubacteria. So let's see what is this Archaebacteria. Okay, now these subgroups we need to learn. Archaebacteria. What are these Archaebacteria? See, these archaebacteria are the most primitive prokaryotes, most primitive bacteria, or we can say most primitive prokaryotes. Okay, these are the most primitive prokaryotes. Archaebacteria are the ancient, very ancient group of bacteria, very old one, very primitive one. Primitive bacteria, or they are the most ancient prokaryotes or the bacteria. Okay, here this ancient bacteria, which is the archaebacteria, this is subdivided into subdivided into methanogens. Then another group is halophiles. Then thermoacidophiles. Okay, so most primitive bacteria, which one is that? Archaebacteria, or these are the most ancient prokaryotes, very old one, very primitive one. And this archaebacteria is subdivided into methanogens, or they're subdivided into three groups like methanogens, halophiles, and thermoacidophiles. Methanogens, halophiles, and thermoacidophiles. Okay, so what are these methanogens? Actually, methanogens means they are like strict anaerobes. They can live in anaerobic condition. Strict anaerobes. Okay, they're strictly living in anaerobic conditions, and they are present in marshy areas. Marshy area. Then in ruminant stomach. In ruminant stomach, you can see this bacteria. They are the methanogens. Methanogens means strictly anaerobic, and they will be present in marshy areas and in the ruminant organisms. In their stomach, ruminant stomach, you can see this bacteria, methanogens. Okay, next comes the halophiles. What is this halophiles? Halophiles, they are anaerobes. These are also anaerobes, and they are present in the salt-rich substrate. They'll be present on salt-rich enrichment with salt, that and all preservation methods to avoid this bacteria, fungal and all. But halophiles are the bacteria or very ancient group of bacteria or which can be grown in the anaerobic condition. They can grow in anaerobic condition and on salt-rich substrate. It's possible to grow such organisms, or these halophiles can grow there. Okay. Then comes the thermoacidophiles. Thermoacidophiles, these are also a subgroup of archaebacteria, and these are aerobes. Okay, these are aerob. They can grow in aerobic condition and they occurs in thermal conditions. Okay, that's why thermoacidophiles. They can live in high temperatures. They can live in hot sulfur springs, then they'll oxidize the sulfur to um the acidic condition, or they can live in acidic condition and high temperature. That's what we need to learn from this. And they lack the peptidoglycan in the cell wall. Usually in bacterial cell wall, peptidoglycan will be there, but in this, they lack peptidoglycan. Okay, this is the main point which you need to know. That thermoacidophiles, they lack the peptidoglycan part in their cell wall. Okay, so this is about archaebacteria. So archaebacteria is subdivided into methanogens, halophiles, and thermoacidophiles. So these characters you may get as questions. Okay. Next, archaebacteria, and one more major group is there in kingdom Monera, which is that eubacteria. Eubacteria is another major group of kingdom Monera. So eubacteria, this is called as true bacteria. Okay, this is true bacteria, and this is also microscopic structure. You know that bacteria are microscopic, and this is unicellular. See, the archaebacteria is a primitive structure or very ancient type of bacteria. Eubacteria are called as true bacteria, like real bacteria, and they are very microscopic and unicellular, and they are prokaryotes. So it's unicellular prokaryotes. And you know that bacteria that may be single, they may be present and in single form or maybe colonies. Bacterial colonies you can see, or it may be a single one. It may form colonies also. And we can say that they are cosmopolitan in distribution. Cosmopolitan in distribution. Okay. So this is the true bacteria. Which one? Eubacteria, microscopic, prokaryote, unicellular, and sometimes it may be single or it can form colonies. They'll form aggregate forms with by forming colonies. And cosmopolitan in distribution. Okay. Next, the features or the structural components of bacterial cell. Structural components of bacterial cell that we need to say: cell wall outside. See, you can see a bacteria. So first one which we need to say, from outer side we are starting. Cell wall is present in bacteria, and this cell wall that is made of peptidoglycan. So bacterial cell wall is made up of peptidoglycan. Note down, this is one question. Cell wall is made up of peptidoglycan. Then plasma membrane is present. Then there will be a glycocalyx layer, which is the slimy layer or the capsule. Around that, a capsule form will be there, or a slimy layer will be there, that is called as glycocalyx, the slime layer or the capsule of that bacteria. Okay, next, the ribosome, bacterial ribosome, which is the 70S ribosome, and the DNA of that bacteria, that will be circular. Circular DNA will be present in bacteria, and they reserve food material. What is that? Food reserve. Food reserve, that is glycogen, lipids, lipid, glycogen. In these form, they'll reserve the food material. Okay, and sometimes, like, different types of bacteria are there, so some structures are present. Structures which are present based on the it is occasional, like based on the type of bacteria, the structures may be present. That is flagellum. Flagellum will be present, or pele. Then plasmid. Then mesosome. Some bacteria will be having that mesosome, and some photos in some bacteria you can see the photosynthetic pigments. Photosynthetic bacteria are there, so those will be having photosynthetic pigments. So these are the occasional features of that bacteria. Okay, so flagellum, pele, then plasmid, mesosome, photosynthetic pigments in the membrane system. That is, depending upon the type of bacteria, it will be present. Okay, so you have to learn the examples for each one, so that and all you will get in detail from your textbook. So this is the main points which I'm saying you need to cover. So with example and all, you need to make notes. While making notes, you need to learn each and everything. Then there are some other microorganisms which are grouped into eubacteria. Other microorganisms are there. And these groups are coming in eubacteria. Okay, so some other microorganisms grouped in eubacteria. We are going to learn about those microorganisms. They are also coming under eubacteria, that is cyanobacteria or blue-green algae. Cyanobacteria or blue-green algae. So the cyanobacteria, they are actually gram-negative bacteria. Cyanobacteria are gram-negative. Cyanobacteria are gram-negative, and they are photosynthetic. See, bacteria, they are prokaryotes and photosynthetic. Which one? Cyanobacteria are photosynthetic prokaryotes. Okay, and they can fix the atmospheric nitrogen with the help of the heterocyst. They'll be having heterocyst, and they can fix atmospheric nitrogen. They can fix the atmospheric nitrogen with the help of heterocyst. Which bacteria? Cyanobacteria. So cyanobacteria are gram negative. Cyanobacteria are gram negative bacteria, and they are the photosynthetic prokaryotes, and they have structures like heterosis, which is helping them to fix the atmospheric nitrogen, and they have one enzyme called nitrogenase enzyme. They have the nitrogenase enzyme. Okay. Examples. We need to learn the examples for cyanobacteria, that is nostoc, then anabina. Nostoc and anabina, these are the nostoc and anabina, these are the bacteria which are called as cyanobacteria. These are the examples for cyanobacteria. Cyanobacteria is also called as blue-green algae. They are the gram negative. Then they are photosynthetic prokaryotes, and they have one special structure called heterosis, which is helping them to fix the atmospheric nitrogen, and they have nitrogenase enzyme. Nitrogenase enzyme, and the examples for which one? This cyanobacteria, anabina and nostoc. They are the examples for cyanobacteria. Next comes the actinomycetes. These are also coming under eubacteria. See, this actinomycetes, they are the bacteria which are forming radiating colonies in culture. These are also coming under eubacteria. And this forms radiating colonies in culture. See, when we are culturing bacteria, these actinomycetes will form radiating colonies. Okay, and among these actinomycetes, especially we can say streptomycete. Streptomyces, one special genus in this actinomycete, one genus Streptomyces. This genus that produces large number of antibiotics. Right, these antibiotics are from bacteria, right? So among these actinomycetes, which are actinomycetes, they are also coming under eubacteria, and while culturing they'll form radiating colonies. And one special genus which is called as streptomyces, they are producing so many antibiotics. From this we are getting many large number of antibiotics. Okay, which one? Actinomyces, which is a genus which is coming under actinomyces. So the genus Streptomyces, which is coming under actinomyces, is producing large number of antibiotics. Okay. Then another one, another eubacteria, Mycoplasma. This is also coming under. This is also a eubacteria. This is also coming under eubacteria. And this Mycoplasma, they are the smallest free living prokaryotes. Smallest free living prokaryotes. Which one? Mycoplasma. It is the smallest prokaryote, and it is a free living prokaryote, and they are also called as what? PPLO. What is the full form of PPLO? Pleuropneumonia like organism. Pleuropneumonia like organism. PPLO. They are the smallest free living prokaryotes, and this Mycoplasma is also coming under eubacteria. Okay. This mycoplasma, actually, they are obligate anaerobes. They are obligate anaerobes and they are parasitic also. They are the parasitic forms. Okay, so they can obligate anaerobe means they can survive without oxygen. In the absence of oxygen also they can survive. They are the obligate anaerobes and they are the parasites. Okay, these mycoplasma, they are insensitive to antibiotics. Okay, insensitive to antibiotics. Which one? This mycoplasma. So here, and they are pleomorphic. That also we need to pleomorphic. Pleomorphic means they can change their shape because they don't have the cell wall. Due to the absence of cell wall, they can change their shape or form. That's why they are called as pleomorphic. They have pleomorphic feature also. They can change their shape due to the absence of cell wall, and they are insensitive to antibiotics that hinder the cell wall synthesis like penicillin. Okay, they are insensitive to antibiotic like penicillin. Why? Because we said they are pleomorphic, they don't have cell wall. They are insensitive to antibiotics which is hindering the cell wall synthesis. Okay, so these are the features of mycoplasma. So here we have learned about which one? Archaebacteria and eubacteria. Under Archaebacteria, we have learned about halophiles, methanogens, and thermoacidophiles. Then under eubacteria, we have learned about cyanobacteria, actinomyces, then mycoplasma. Okay, they are the eubacteria, the true bacteria. Archaebacteria are the ancient or old or primitive type of bacteria. Okay, then we learned the general features of kingdom Monera also. See, now for you it will be easy to make a note of this kingdom Monera, right? So kingdom Monera now you can get easily, and you can get almost all the questions from this area. So this is how you need to learn the general features and the classification or the groups or subgroups in that. With example you need to learn, so that you are covering the 80% of that portion. Then some questions, some applied type questions will come from some portions, so that you need to think and write. Okay, so now we have covered kingdom Monera and what is biological classification, what is five kingdom classification, and among that five kingdoms was first one, which is that kingdom Monera we have learned. Okay, and in the next class we'll be learning about the other kingdoms also. Then we'll move to the question session. Okay, thank you.